I'm Alexis Johnson. I'm the social media manager here at RCSA. I'm joined by Jim Broach, Executive Vice President and Director of RCSA, and Apkar Apkarian, PhD, Professor of Neuroscience and Anesthesia at Northwestern University. He is our first Facebook Live guest for 2023, and we are very excited. A little bit about him. The past 35 years of Dr. Apkarian's life have been devoted to unraveling brain mechanisms that underlie acute and chronic pain, and more generally, how the brain dynamically processes information that gives rise to perception. Over the last 20 years, he has used brain imaging technology to delineate brain biomarkers of chronic pain with an emphasis on brain mechanisms of chronic back pain. These efforts have been quite successful, and his work has resulted in many significant advances in the field of pain research, which include the first identification of gray matter atrophy related to chronic pain, the first account of brain activity unique to spontaneous fluctuations of chronic pain, the first characterization of resting state brain network abnormalities in chronic pain populations, and many more. These observations have been replicated across multiple international laboratories. With over 30,000 citations, his work has been continuously funded by the National Institutes of Health for 3 decades from 6 different institutes within the NIH. This work would not have been possible without the enthusiasm, creativity, and persistence of a large number of graduate students and postdoc fellows whom he has trained, as well as the talent and inspiration of his colleagues. We are very excited to have the doctor here with us tonight. As always, remember that while the information shared here is helpful, please consult your physician for personalized medical advice. Now, I have a few announcements before we turn it over, but Jim, I know you have a few as well.
Thanks, Alexis. Take a breath. Basically, I want to say that we first focused on his work in 2010. We had an article in the newsletter. And now 13 years later, he's back. And I'm so excited for that. But at the end of this month, what is it? It's Rare Disease Day. And I have 2 dogs, and they're going to be wearing Paws for a Cure. And I hope that you go to our store and you'll outfit your service dog or your pet therapy dog or just your best friend and that. And I'm real excited about that. And also, we ask you come to our social media and your hashtag. Yeah, share your stripes. All right.
There you go. Definitely take pictures in your orange and share them on social media, just like you would do for CRPS or Color the World Orange. So definitely do that, and definitely share your stories on social media. We will definitely post more about that on all of our channels. And a couple of other announcements. Our Young Adult Weekend, which has been rebranded to Young Adult Retreat, is happening in Philadelphia from June 2nd through June 5th. So those details we posted on social media, so be on the lookout for that. And we also have multiple Facebook Lives happening this spring. We're super excited to get back to it. After this one, we'll have one on March 2nd and then March 23rd. So once again, keep an eye on our social media channels and in rare form, which of course goes out monthly. Tons of announcements, tons of information in there. So we're excited to continue to provide that for you all. But enough for the 2 of us. You all see us all the time. I'm going to pass it over to tonight's guest. So Dr. Apkarian, the floor is yours.
Thank you, Alexis, and thank you, Jim. And thank you for inviting me to this venue. And I'm happy to discuss some of our research over the last numbers of years. And I do want to share with you some data. And so let me get my slides and share my screen so that I can talk about the data that I would like to discuss with you. And we start that up. So, and I assume, is this okay? Am I sharing the wrong screen? Let me see. Let me do this. Give me a second. Sorry. I guess. How's this? I assume this is better. So, yes, as Alexis just mentioned, I have been studying human pain conditions for many, many years. I'm grateful to all the patients who have accepted our invitation to be part of a whole series of clinical studies, basic science studies that we have been doing over many years. And today I want to try to, in a sense, distill some of that work into main concepts that I can share with you, and perhaps that would help you understand what is chronic pain in general, and or what is CRPS and reflex sympathetic dystrophy conditions, and at least how we envision the condition ourselves. And let me see. So I will try to summarize our basic findings, and of course, our findings are all embedded in the neuroscience of pain. In a sense, I have been pioneering and continue to work on what happens in the brain when someone is developing chronic pain, what processes in the brain are in fact predispositions or characteristics that make people develop chronic pain. As you may or may not know, in general, many people with similar injuries, similar types of events, some people develop chronic pain, others don't develop chronic pain. And how can we untangle all of that from the role of the brain in pain? And in a sense, I want to start with a few definitions of pain and the brain circuitry around it, so that we can speak the same language. Let me move on and say, okay, so very simply, of course, we all know acute pain is not the same as chronic pain. Acute pain is a sensation. You burn your finger and it hurts for a few minutes and it goes away. The acute pain controls human behavior. When you have pain, you withdraw your hand from that painful stimulus, your heart rate goes up, your attention goes up, but then in a few minutes, it all goes away. In contrast, chronic pain is pain that the standard definition would say is pain that persists after the injury and the healing process has happened. So in that sense, the site of the pain in chronic pain is going to be more centralized than the site of the injury itself, almost by definition. Okay? And we'll come back to this idea, probably a number of times. Of course, the world population continues to increase in the rate with which chronic pain is reported. It has a huge cost on society and on quality of life. And by and large, we don't have specific treatments for chronic pain. We have management drugs, management procedures, whether it is drug therapy or multidisciplinary therapy. All of those are efforts to alleviate the burden of chronic pain, but we have yet to uncover treatments that are specifically for chronic pain that can cure the pain. And what I would like to sort of talk you through in my discussion is that we are at the brink of getting there. So I am excited with the research we have been doing, and in a sense, I think we are at the edge of, in fact, discovering very specific treatment strategies for chronic pain, and all of that happens by studying the science of chronic pain, by uncovering mechanisms of chronic pain. And by and large, I will continue to emphasize the role of the brain in all of these. So I will show you some data of what we have achieved over the last few years, always with the intention of also giving you hints of what our clinical studies are going into, in a sense, to discover new treatment options for chronic pain. Okay, let me move on from this slide. And so, again, some simple definitions that I would like to talk about. Pain is what you report. Pain is in the brain. The pain is the suffering that the subject has, whether it's acute or chronic. It is a subjective perception, right? And I would like to also distinguish it from the nociceptive inputs. Nociceptive processes are neurons that can give rise to pain perception, but they don't have to necessarily give rise to pain perception. And in a sense, pain is a conscious signal, while nociceptive processing happens in the periphery, in the spinal cord, in many different areas of the brain, but the brain can suppress those things or exaggerate them. But when you are in conscious state of pain, of course, nociceptors are involved, but that does not mean that those nociceptors necessarily give rise to pain perception. Just a nuance that I want to share with you. Now, I have been studying the human brain, in fact, the human and animal brains for more than 30 years. And this is a very simple sort of cartoon diagram of what we can do with brain imaging. So we can study the brain anatomy. This would be the gray matter properties. The gray matter are the brain areas where the neurons, the cell bodies, are located in the brain. We can study the axons in the brain, what neurons are connected to which neurons in the brain. So those are the connectivity, the information transfer from one area to the brain to another. So we can actually study the axonal integrity of the brain. More recent studies, in fact, have started looking at the glial properties of the brain. These are basically support cells in the brain, which are becoming more and more important in our understanding of pain and in neuroscience in general. And in a sense, looking at the glial properties, we are looking at brain inflammatory processes that can happen. So those are structural properties. Numbers 1 and 2 are structural properties that we can derive from half an hour of scanning of the brain of any subject, right? Then we can also study the brain as activity, the physiology of the brain. This would be functional magnetic resonance imaging. And here I have two different cartoons. Number 3 is if I give a painful stimulus to a subject, the heat, let's say that for 10 seconds and on and off, and then we look at what brain areas are activated. This is the brain picture. Now the color blobs here are the brain areas that are responding to that stimulus. So in a way, this is a nociceptive signal that the brain is reflecting, and these brain areas, we can think of them as being involved in the perception of the pain. So, and again, here the red line, the wiggly red line, is in fact the brain activity from one of these regions superimposed on the stimulus. So you can see that how the brain activity goes up and it goes down as we have a painful stimulus, as we heat the skin up to a painful intensity and then bring it back down, and the brain activity goes down. So this is activity so we think of this as reflecting local neuronal activity in the brain in relation to the specific task, in this case, to a painful stimulus. So this is, in a sense, brain activity related to the acute pain perception, right? As every time the stimulus goes up, the subject reports pain, goes down, the pain is gone, goes away, and these regions are reflecting that activity. Now, so this is the neuronal activity, the physiology of the brain that we can study. Number 4 is how the brain communicates physiologically with each other. So what brain regions talk to each other? And in a sense, we can, rather than looking at activity, we can look at information transfer between regions. And that information transfer, we think of them as a network of the brain, as connectivity, and the properties of these network properties are, in a sense, another order of us understanding what the brain is doing. In this case, which brain areas are sharing what type of information with each other. And that information sharing is is a massive process in the brain. It's happening all the time. So the brain has both neuronal activity and sharing of neuronal activity across brain areas. So these two panels, numbers 3 and 4, are complementary to each other, and one cannot happen without the other. But the properties of each one can give us different insights about the brain, right? So these are essentially the 4 dimensions within which we can study the human brain. And we've been studying those in a variety of chronic pain conditions. Okay, so before I start showing you the data, I also want to simplify my talk to primary concepts, right? The brain is a complicated structure. The cortex itself has 80 billion neurons in it. Those 80 billion neurons have 140 trillion connections with each other. And so our understanding of the brain remains rudimentary. Don't believe what people say in the social media. We are still very far away from understanding how the brain really works. And so in that sense, we need to make progress. We need simplified concepts. So for today's discussion, I will take this very complicated structure and discuss two parts of it. The neocortex. The neocortex is the surface of the brain. This is the area of the brain which is most elaborated in humans, right? This is where we think the humans distinguish themselves from other animals, and this is where we do our most complex conscious processing. So conscious perception, we think, is necessarily involving the neocortex. If I take the neocortex out, that animal or subject will show no conscious behavior. Underneath that cortex is this limbic brain area. The limbic brain area is a whole series of, again, brain circuits, neurons, and connections that fundamentally are creating the coloring of your conscious perception. In a sense, they are modulating your emotional experiences. So the neocortex receives inputs from visual, tactile, olfactory, auditory inputs, and starts processing it at the neocortical level, while the limbic subcortical areas interact with those signals to interpret it as a happy scene or a sad scene, or a suffering scene or a suffering perception, right? So the interaction between the limbic brain and the neocortex really creates the emotional color of experiences. And so I will try to limit my talk to these two concepts and how these two concepts are interacting in acute and chronic pain. Okay, very simply, if I look at the brain activity for pain, this is a meta-analysis of something like 3,000 studies of pain. You can see a very robust set of brain areas that are activated. Okay, I'm sorry. The panel on the left is for vision, the panel on the right is for pain. So if I look at 500 studies for pain, I see this red colored areas of the brain that are activated. And if I do the same analysis for vision, I see another set of brain areas. There is no overlap between them, but we also see that if I can actually look at the highest activated areas, that I have a very high confidence that there are some brain areas that are necessary for vision perception that are not involved in pain, and necessary for pain perception that are not involved in vision. So that's nice. So we can sort of dissociate the brain into cortical areas that are necessary for different perceptual states. And fundamentally, we have a brain signature that shows us what brain regions are involved in pain perception in general. Okay, let me move on. Okay, so now the next concept that I want to also emphasize, and I'm going to show you some data about it over the next set of slides, is that chronic pain is not the same as acute pain that does not go away. Okay, so what do I mean by that? In a sense, what I mean is that when we look at the brain anatomy and the brain properties of different chronic pain patients, their brain anatomy is not the same as subjects who are in acute pain. Okay, the brain anatomy, the brain properties are all changing in time in chronic pain. And different types of chronic pain show different brain properties. So the interaction of the brain with pain is both changing the brain and defining the pain at the same time. So, and what I would like to show you some evidence about is that there is both a predisposition and a reorganization that happens with chronic pain. The predisposition, in a sense, are the brain properties that increase the risk for the same type of injury for someone to develop chronic pain versus another. So those are properties that are, that are coming with the subject, and it does not matter, and it does not really reveal itself until an event happens, while the reorganization is a consequence, right, of living with the pain, its condition itself. Okay, so coming back to this slide where I was showing you these 4 different ways of analyzing the brain, what I want to emphasize is across all 4 modalities of brain analysis, we see abnormalities of the brain properties in chronic pain. So brain gray matter anatomy changes with chronic pain, the axonal connectivity changes with chronic pain, brain activity patterns are changed with chronic pain, and brain functional connectivity, functional network properties, information sharing in the brain are distorted with chronic pain. And in a sense, all of these things together define a chronic pain state. Okay, very briefly, let me just go through this slide. It's a complicated slide, but don't get too impressed by it. This is a drawing that René Descartes did back in 1644 with the concept of pain perception. Basically, there's a fire, the fire sends a signal to the nerve transmits that information to the brain, and the brain feels pain. On top of this, what I'm trying to show is that, in fact, at all levels of the nervous system, being at the periphery, at the level of transduction of information, at the level of the spinal cord, and at the level of the cortex, with chronic pain, we see reorganization of the system. And in a sense, there's a lot of information known about what happens at the site of injury. There is a lot of animal studies of those events, and a lot of animal studies of the spinal cord sense reorganization with pain. And now, some 20 years ago, so we pioneered the idea that both the brain anatomy and function are also different. So then the question is, where is the chicken and where is the egg? How can we unravel all of these events that are happening in the organism, and can we create a scientific concept from which we can learn and derive some new ideas that can move the field of pain management into a more productive place. So, okay, sorry, this is now my overall outline of my talk, basically that we can find biomarkers. This is basically brain properties that are both predictive and a consequence of chronic pain in humans. And we can, from those concepts, we can go back into animal studies and show that the mechanistic processes underlying these things, and also then show that fundamentally the brain's limbic emotional properties, the inner part of the brain that we discussed earlier, are causally controlling the chronic pain state. So they both present as risk factors. The limbic emotional brain properties are fundamental risk factors to develop chronic pain, and they are the ones who are controlling how the cortex is reorganizing in the transition from acute to chronic pain. I also want to show you some data that, in fact, these concepts, in fact, do give us mechanistic ideas, and they are pointing to new treatment opportunities. From a temporal viewpoint, we can think of, a pinprick or burning of the fingertip as an acute pain event, right? Now, if that acute pain event starts to persist over time, we can think of this as a subacute. And then if it does not go away after the healing process has happened, then we will call it chronic pain, right? So within this timeline, from acute to chronic pain, we can think of different processes that are happening. First of all, again, predisposition would be properties, even before the acute pain happens, that increase the risk for chronic pain. The predisposition, together with the injury, then starts the process of traversing from acute to subacute place. And then we think of this transitional state, which would be a time window within which the brain, the nervous system, is reorganizing either to become chronic pain or for the pain to go away and develop the new brain state. Now, the traditional view is that all these states are essentially driven by spinal cord and nerve fibers coming into the spinal cord. The work we have done over the last 20 to 30 years now has a very different concept. And this is the general idea that I want to emphasize. Again, the brain emotional circuitry, the limbic emotional circuits, define the risk. So number one, the predisposition is the risk. And emotional learning circuits are the transitional state, right? During this transition, whether this circuitry reorganized one way or another, we end up with either in chronic pain or the pain goes away. And when we are in the chronic pain state, number four, there is a new brain state with distinct anatomy and function. So this is the general big picture, and I'm going to show you some evidence along these lines over the next slides. Let me just skip over this slide. It's not very important. Now, most earlier studies, even our own studies initially, was the simplest way. How do we study these things, right? We bring a patient who has chronic pain. We look at their brain properties. So in a sense, we are studying them in their maintenance state, right? They're in that chronic pain state. Then we compare their brain to healthy controls and say, what properties are different between them? And I'm going to show you some data along those lines. This was the main, it remains, in fact, a big ongoing important approach to understand, what are the properties of the brain in different types of chronic pain, right? And very simply, again, going back to that overall picture for acute pain, this is the overall brain activity for acute pain. Now, if we look at a group of chronic back pain patients and identify the brain region that's directly related to their back pain, we see a very different picture. We observe that brain regions that are not in these areas are being activated. And again, this is part of the limbic emotional circuit. And in a sense, this is the first hint that acute pain and chronic pain show different brain activity patterns. All right. And I don't want to go through all this detailed data, but we can dissociate acute pain and chronic pain in the same subjects by doing multiple control data analysis. So that simple fact that we can observe, let me go back here and see that this medial frontal cortex, a region that's tightly related and that controls, oops, what happened? And controls emotional activity in the brain, is uniquely activated in chronic pain, gives us the idea that we should look at treatment options along those lines. Okay, my slides are jumping around, I don't know why, but I'll control them. Here we go. So this is now the left panel is an experiment we have done in rats. And basically in these rats, we give them this drug, it's called D-cycloserine, over a month and a half. These animals have chronic pain. We have given them a partial nerve injury, so they are highly sensitive to stimuli on that limb. Very similar to CRPS type of pain. In a sense, the limb is very sensitive to external stimuli. In fact, the sensitivity is so high that on the injured nerve, on the injured part of the limb, stimuli that are in the range of 0.1 gram, the animal will retract away from it. The uninjured limb, 12 grams of force is the amount of force that would make them move away from that injury. So this is their hypersensitivity. This is their tactile allodynia on that injured limb. Now, if we start giving them this drug over 45 days or so, you can see that sensitivity is decreasing. The amount of force needed for them to respond is getting to be higher and higher. The control condition, they're still just as sensitive. And then we stop the treatment, and it takes another month or so, month and a half, for the effect of that treatment to go away. So this is, in a sense, a drug treatment that's taking many weeks to exhibit itself. So, and we think it is reorganizing the chronic pain brain into a more normal state. And as a result, when we stop the treatment, that effect is still there for another month. Okay, so this was a drug that we initially tested back in 2007. This paper was published in 2007. 10 years later, we completed a very small human clinical trial. This human clinical trial, again, you can see a similar pattern. Let's just look at this change in pain in panel B here, between D-cycloserine versus placebo. You can see in time, the D-cycloserine the study was only for 6 weeks. The D-cycloserine is decreasing the pain more than the placebo. So, and we are, in fact, I wanted to show you this slide because this week we completed a phase 3 trial with this drug for, again, chronic back pain patients, where we have actually studied 170 patients. I don't have the results, but we will know the results in the next week or so. And we are hoping that with this drug, we can literally cure people from chronic pain, at least for multiple months. We don't know if the result will work or not, but we are at the edge of finding that result in the next week or two. So I'm very excited with these results. It's taken us about 15 years to get here, right? So human studies take a long time, and human clinical trials for drug research take even longer. Let me move on and just show you some other data. So this is a study that we did, again, 20 years ago or so, basically showing that, in fact, the brain gray matter is changing with chronic pain. now there are many other studies like this for different types of, was for chronic back pain, this is for fibromyalgia, this is for tension headache. In each one of them, we see different brain anatomical changes. This is for irritable bowel syndrome. And the nice thing that we can think about is that these changes seem to be at least partially reversible. And here's a study by Dr. Seminovitz, published 10 years ago or so, in, again, back pain patients who went through surgery, and they actually studied patients where the pain was relieved. And what you can see is that is a reversal of the brain anatomy when the pain goes away. So with the proper treatment, when we have the proper treatment, the brain properties are at least partially reversible. Okay, so now let me just go back to this cartoon, of this transition from acute to chronic pain. And again, some 10 years ago or so, we did this crazy experiment or study where we bring in back pain patients who are very early on in their back pain, and we studied their brain multiple times over a 1-year period. And basically, we are trying to find predispositions in their brain, essentially brain properties that predict who 1 year later will or will not have chronic pain, right? So everybody enters into the study. Some of them develop chronic pain, others just get better. And then we say, at this time period determines this bifurcation, and that becomes a risk factor. Let me just move on. This is and so actually this is a very simple concept I want to share with you. Again, now if we simply look at the brain activity of these people at the subacute back stage, the brain activity of all of these people are similar, and they're mostly showing cortical activity. Over 1 year period, the people where the pain disappears, there's no more brain activity, but that's good. While the people where the pain persists, it shows up in all of these limbic brain areas and very little activity in the cortical areas. So in a sense, in the same subject, the pain perception changes its brain circuitry over time. Okay. And again, the brain activity as a network, we can identify it as the limbic brain, limbic cortical connectivity. And this limbic cortical connectivity becomes a risk factor and a predictor of chronic pain. I don't want to show you all the details, but we can see that the simple amount of information shared between two brain areas predict at time 0 who will have chronic pain 1 year later. And this is, in a sense, the predictive accuracy. We're about 80% correct with this single number to show that 1 year later, who is at higher risk to develop chronic pain. And now let me just move into some animal data. In animal data, the nice thing is that we can literally study this transitional state, because we give these animals an injury and look at how things are changing. And we can look at similar properties derived actually from the human data that we now test for the same circuits at, of course, at much higher resolution and much more invasively, right? I don't want to show you all this data, but all I'm showing here is that the brain anatomy, the single neuron anatomy changes with pain, local brain activity changes, and local neurotransmitter levels change with chronic pain. And these are all brain limbic areas that we're looking at. And in fact, we can control this neuronal activity with, optogenetic technology. The details don't matter. But if I change the excitability of these neurons in these limbic areas, I can either decrease or increase their tactile allodynia, how much that paw feels painful, with simply changing the excitability of neurons very far away from where the injury was. Okay. And I also want to emphasize an important point. We can do this for many areas in the limbic brain, in the sense that the brain is highly interconnected, and controlling one area will control other areas. So it actually becomes a very complicated circuit, or in a sense, you can control one area. If another area is not controlled, then you still have the pain perception as well. All right. Okay, and so regarding this transitional state and all of that brain circuitry that I showed you that we have analyzed gives us, again, another clue of another drug treatment that we have been studying. And this is a combination drug, dopamine plus an anti-inflammatory drug that we have been studying recently. And let me show you some of that data. This is, again, in human studies. So this is now in some 65 patients, chronic back pain patients. We have given them either this combination drug, again, for 3 months, and then we observe their pain. You can see, so here we have the drug versus placebo, and both males and females. Overall, there is a decrease in pain. And the nice thing is, again, we think we are looking at a treatment where after we stop the treatment, the effects persist for many months. Okay, and in this case, the best effect is the treatment in females. In fact, the female participants use less of the drug and get better pain relief with it. And the pain relief is directly in proportion with this brain connectivity in these patients, so that the drug effect, we think we can see that it is in fact acting through the same circuitry that we have identified both in humans and in animal models. Now, this is a very small study, limited data, and the results remain, suggestive, but not conclusive. We just sent in a multi-million grant proposal to do this at a much larger scale in a much larger population of subjects, a phase 2, phase 3 clinical trial that we hope to get started very soon. So let me just move on. So I'm trying to essentially conclude my talk. Again, this is the 4 phases of the transition from acute to chronic pain. And there are multiple, questions that remain to be answered. So, we have a whole variety of chronic pain conditions. What are the differences between the risk factors, for example, between different types of chronic pain? And how are these different circuitry, contributing to these risk factors? And also how the inputs, the injury-related inputs and the interaction with risk factors vary with different chronic pain conditions. Those are all questions that we don't know the answers to, but we hope that if we start learning these details, we should be able to derive very specific chronic pain type dependent treatment strategies. So overall, what I want to say is that there are multiple brain properties that are risk factors. We don't know the details of them as a function of the chronic pain. Their interaction with the limbic emotional brain and its interaction with the injury creates this transitional state, which, when it becomes chronic, defines a new brain state. And let me show you in a more cartoonish way. So here is, at the bottom is this limbic brain again, and at the top is the neocortex. Nociceptive inputs are coming to both of them all the time in our daily life, right? Now, if there is a big injury, this nociceptive input gets larger and one starts feeling pain. On the other hand, if the corticolimbic properties are such that you can suppress the limbic emotional responses, then the pain will go away. if that does not happen, if you activate this emotional circuitry, then they in turn will send signals to the cortex, an emotional learning signal that will reorganize the circuitry at the cortical level and create this emotionally driven cortical state of being in chronic pain. So let me just conclude with some main concepts that we think we have established now for multiple years. Since the brain properties are for a very large portion of the risk for chronic pain is based on brain properties, we should think of chronic pain as a neurological vulnerability. So, in a sense, both genetic, nurture properties predefine people's risk for developing chronic pain, and which may be uniquely different for different types of chronic pain, right? And all of that is fundamentally determined by this limbic emotional circuitry as to providing the risk factors. And their interaction with the injury is going to define how the pain then is developed. And most importantly, translational studies between the human brain imaging studies, animal mechanistic studies, and their translation back into clinical trials it gives us this general mechanistic ideas from which we can derive treatment concepts. And in a sense, the overall idea would be that chronic pain is a state that the brain becomes more and more amplifying the nociceptive signal, and we think of this as creating an addicted state of nociception that the brain is generating, and we need to reverse that to go back to a normal state of beingness. So many students, faculty, friends have contributed to our research, and I should thank all of them. And we have had many years of funding from different, especially from National Institute of Health, to do this research, and I want to thank them as well. So I'm going to stop sharing my screen and see if there are any questions that I can answer, and we can have a discussion.
Oh, yes. Thank you so much for that. I'm actually going to bring Jim back on. I'm like a little rusty at Zoom. It's been too long. Hold on. All right. Jim, bring you back on. All right. Thank you so much. And actually, Jim, I'm going to put you in the hot seat. You want to start off with questions?
I'm always intrigued with CRPS, that the same particular injury or accident could occur to one individual and they develop CRPS, and the next person, same thing, not. And when you talk about predisposition, do we know anything more about that?
Unfortunately, we don't know much specifically for CRPS. And the problem is, in a sense, partly I am directly to be blamed for it, because 15 years ago, as you mentioned, we were in fact trying to do brain imaging studies in CRPS, and basically it was very hard to do the studies because we could not recruit the patients. We did not have large enough patients participating in these studies, so we literally had to switch back to back pain, which is much more common, but I have to emphasize, even in back pain, we don't have enough patients in our studies. We're always struggling getting subjects to participate in our studies. And I understand, these are quite, burdensome studies. We do all kinds of brain imaging. So I also thank all the participants in doing these things. But so that becomes a really, limiting as to how much we can do and how much we understand. I am very much interested in pursuing the CRPS population, but we just simply don't have enough access to patient groups to do these large-scale, long-term studies that take many years to do, and that takes participation of many subjects into these studies.
I appreciate that. It's another consequence of being a rare disease.
Yes, exactly. Yeah, exactly. Yeah.
So we have a question that was earlier in the presentation. So hopefully this makes sense for where we were or where you were during your talk. But the question is the new brain state with chronic pain the same for type 1 and type 2 CRPS?
We don't know, is the reality. I suspect they're different in a sense that every chronic pain condition that has been compared to any other chronic pain condition has shown unique brain reorganization. And in a sense, the reorganization is basically the brain trying to cope with the condition and to adapt with the condition. And different types of chronic pain, by definition, are going to have different signatures behind them.
All right, so we have a lot of questions about trials and people want to know how they can participate, what cities are they in?
Well, I'm in Chicago. Send me an email. We would love to have you in here, but we cannot fly you here, and we just don't have the resources. Also, our studies are not pharma company funded. These are basic science studies. They are driven by NIH dollars, which are always very miserly and limited in their amounts. Although they have funded us for many millions of dollars in the past, so I should not complain too much. But our abilities are limited, but we love to have patients volunteer and participate in our studies. Please contact me. I'm happy to put my email address maybe on the chat box or something. Yeah.
There was a great surge of hope when you mentioned the data being sent to be analyzed regarding the medicine that is, you said the word cure for chronic pain. Could you just elaborate a little about that?
About what? What was it?
About the trial that you completed and you're really hopeful that
Yeah, so this is a trial that in fact was funded by Department of Defense. It's a multi-million-dollar project. We've been studying about 200 chronic back pain patients over the last six and a half years. We just closed the study. So of 171 patients were randomized into the study. Every person who enters the study gets either placebo or drug for 3 months. Then we stop the treatment. We follow them up for another 3 months. And as I showed you with the animal data, the concept is that if a person responds to this drug, their pain will continuously decrease over time, right? And then when we stop the treatment, they will stay at that level for the next 3 months. That's the concept of the study. It's blinded. I have no idea what the data is going to show. And yes, I'm in high anxiety in a sense that in next week, we will come to the conclusion of 15 or 20 years of my research, whether it will succeed or fail. It's just that simple. And so that's where we are. So I wanted to share with you the pathway that we travel in these things, right? It's a very long path. I mean, many students, faculty, NIH grants have created this whole road. And at the end of it, it's like, we throw the dice and we see what happens, right? And of course, if it's positive, I think it will have a very big impact on healthcare in chronic pain in general, but the chances of it being positive are very small. I know because in the last 30 years, not one new drug has been discovered for chronic pain. So the odds are completely against us, but we have to take the risk and do the studies, right?
well, I see the clouds. Got that.
All right, a couple more questions. So this one also came in a little early on. It's asking about thoughts on physical therapy and chronic pain. How much is too much?
Okay, I have strong opinions on the topic. Physical therapy, I think, is great. Physical therapy can always be too much. You need to pace yourself and don't listen to the therapist, listen to yourself. The best guide is self-guided, I think, therapy, especially with physical therapy, right? You should not make your pain worse. It's just that simple. But you should also fight against the pain. And in a sense that physical therapy, cognitive therapy, behavioral therapy, massage therapy, all of those things, I should make the message, I should emphasize, engage your limbic emotional brain. And in a sense, they are all helping you fight against the reorganization of your cortex that has happened. And if you can use those tools to fight it, you should go ahead and do it. And I'm not going to advocate any one procedure over another. They all can be helpful, and obviously it's a personal choice, and what is available and what is doable are all practical issues that one has to deal with. Yeah,
It's also what's paid for.
of course, yes, financial resources as well. Yeah.
All right, another question just came in, and I feel like I'm going to butcher one of these words. So, has your research determined if it's possible to break the brain's addiction to nociceptors, and would that depend on how long the chronic pain has existed?
That's a great question. We don't know the answer to that. Again, I mean, those are great questions, but they're very hard to answer, right? It would take 10 years to answer that question. Now, let me qualify that. We think that transitional state that I was talking about, the transition from acute to chronic, that state is a critical time window. And we define that between 3 to 6 months from the initial injury. And within that time window, I think aggressive treatment will give us better results. This is what we are trying to study with that dopamine drug that I told you about. And in a sense, we are targeting that time window to see if we treat people properly in that time window. After that treatment, they simply will not go back to develop chronic pain. So there is a window within which I think aggressive treatment is going to be good. Unfortunately, again, the amount of data that we have within of that concept remains limited, and there's more evidence that needs to be generated for it too. But once it becomes 1 year, 2 years, 3 years, I don't know what the probabilities of reversal end up being. I mean, if our drug study is positive, I think it will start giving us hints who gets better results versus who doesn't, and whether that is dependent on the number of years different subjects have been living with their pain or not. Those are parametric studies that we will explore if we have a positive trial.
Jim, do you have any more questions?
I think we just have to emphasize that this has been recorded, slides will be forthcoming. And that I really thank you for coming and sharing. Everything is in the brain, and it's
And I think that's what makes it so challenging. Someone asked, for CRPS to be known since the Civil War, why is there still so much little information about it? And I think that kind of answers it.
Yeah. I mean, it's a complicated field. These are hard studies. They take many years to do these studies. And yet I hope I gave you a positive message that in fact we are making roadways, we are understanding the system, and the understanding is giving us new ideas of how to treat the conditions. And that's the bottom line. Science has to give us new pathways of treatment, and I'm hoping we are at the threshold of one of those, in a sense, right?
and I think we really need to talk about too is that of all medical research, pain is the stepchild. It really has been less than 2% of all healthcare research dollars are invested in the number one reason you go to a medical office.
Yes, but the good news is NIH and Congress have realized that, and those dollars have increased. and NIH is putting a lot of effort into making large-scale aggressive trials to break that cycle and to come up with new treatment options and new mechanistic ideas of these systems. And in many ways, I think some of our ideas were the pioneering ideas that now NIH has adapted and is pushing forward at much bigger scale than we could ever do ourselves in a single lab with a few students, in a sense, right? Yeah.
Thank you for helping me be more positive.
Yeah.
We do have one more question, and then I will let you go. The last question for the evening, and I'm looking for it, but I know what it was. I know you had opinions on, physical therapy and whatnot, but how do you feel about spinal cord stimulators?
I'm more skeptical of them, actually. Although I also need to admit that there's a whole new generation of them. They keep advancing their technologies, and I have not carefully looked at them. But there's also no question that there's a huge placebo effect in these gadgets. On the other hand, if it works, it works. There's no one should not. I'm a basic scientist, and I keep wanting to think about the mechanisms, and that's the only thing that gets me excited. But when there is efficacy, we just take it, right? There is no questioning someone who responds to the treatment. It's just that I'm a bit puzzled with the cost of these things and being sold as like aspirin in a sense, mostly because it's great revenue for the people who are selling them, and I don't know. I am conflicted with them. Yeah.
I have the adage of what John Lennon said: "Whatever gets you through the night."
Right. Of course, absolutely. On the other hand, each one of these things is a $40,000 pop, right?
Exactly.
Yeah.
And what we're discovering now, and especially anesthesiologists, don't want to do medical management; they just want to implant now, and it's sad.
Exactly. Yeah.
Well, thank you so much for all this great information. To everyone watching, of course, we will have this video pinned to the top of our Facebook timeline for easy access until our next Facebook Live, and we'll also add it to our YouTube channel. So if you want to rewatch it, you will have the opportunity to do so. And speaking of our next Facebook Live, join us in early March as we'll chat with Cynthia Toussaint and John Garrett, who will discuss the journey of having CRPS and cancer. I think it is a talk that many will find interesting and inspiring at the same time. So more information about that will be posted on our page, all of our social channels, as well as in Reform and our newsletters. I hope you all are subscribed. So if you have any questions for us, if you need a support group, if you're looking for a new physician, or if you want to tell your story on our blog, please send us an email at info@rsds.org, or you can send us a message here on Facebook. Thank you so much and have a great evening.